up the lesson for the moment now and make sure that everyone is here. I also want to remind everybody that you can also put questions in the chat as well. I can see the chat. That's just my little reminder that the revision lesson is on the way. Okay, I'm just going to do that. Sangeeta, I just saw your question about blood groups. There is a page on explaining blood groups as far as I remember. I will double check that for you just to make sure it's in the cheat sheet, but I'm pretty sure it is there. I'm going to wait just like 2 more seconds for everybody to join. And then I'm going to swap over to the things that we are going to cover today. Remember that I'm going to cover a majority of things that are in the June exam for the majority of the current matrics. And then also, if some of you are people who are rewriting your exams and you're joining us for this lesson, this is going to be like a mixture of things. I'm going to do some paper 1 questions and some paper 2 questions. So just be aware that you can use this video to revise those things. We're of course going to have more lessons together. I'm so sorry that I've had to reschedule this one. It's just because of load shedding and not having enough battery power to be able to do it. But I think let's get into the lesson, everybody. So I'm just going to swap over to the questions for today. And I'm going to go forwards and I'm going to start with this one. Okay, so what I thought was most appropriate was we're going to ease into the lesson and I'm just going to do some multiple choice questions. Now, for some of you, you're probably like, oh, those are so easy. Why is she starting with that on a live lesson? The funny part is I actually think a lot of us get let down by our multiple choice, considering that each one of these questions is for 2 marks. You shouldn't be getting any of these wrong at all, right? So let's get into them. And these are going to be some paper 2 questions because these are all genetics. So a lot of you have done genetics recently. So let's break down the questions. It says the diagram below shows the pattern of an inheritance of a disorder. So we know it's a disorder. It's a very simple diagram. We've got male who is not affected, female who's not affected, and then a female who is affected. So now we've got to break this down. And it doesn't say it's a sex-linked disorder. It doesn't say it's a non-sex-linked disorder, nothing like that. You've actually got to figure that all out for yourself. I'm already going to say that it's not sex-linked. And the reason being is you can't have a father who doesn't have the disorder and then a daughter who does have it and then be sex-linked. In other words, if the daughter has it, the father must also have it as well if it was sex-linked. So this is definitely autosomal, but if we actually look at the question now, that might not be too important for us to figure out. But let's have a look at the question. It says, one can conclude that the disorder is caused by a recessive allele with both parents who are heterozygous, a dominant allele with both parents being heterozygous. Now remember, being heterozygous means that you are, and I'm just going to use the letter T, you are a big letter and a small letter, right? So that's what heterozygous means, right? So they're saying that either of the parents could be that. The next option is it's a recessive allele where one parent is homozygous recessive, which means that they would be 2 little letters, while the other is big letter T, small letter t, right? Next one, dominant allele. One parent is heterozygous, so big letter, small letter, while the other is homozygous recessive. Okay. So now what we have to figure out on this one is this inheritance a dominant or a recessive? Now, remember, if it was a dominant disorder, you only need 1 capital letter to get it. Now, if that's the case, right, that means that, let's just play the game here and fill it in. That means that this girl over here has it, the capital T. That's just not possible because then both of her parents would need to have 1 in order to give her 1 each, right? So then they would also have the disorder. So already we can just discredit that because we know that that's not the answer. So all of these that say dominant allele, we're just going to cross those out. Those are not our options. And we're going to focus on the other 2, A and C. I do believe this is a recessive disorder, which means in order to have it, you need to have 2 small letters. So I'm going to go in here confidently and say that this girl is 2 small letter T's. And now I'm going to have a look at my 2 options. So I definitely know it's one of these 2, A or C, but now it says where both parents are heterozygous, or where one parent is homozygous recessive while the other is heterozygous, right? Now, I'm going to say the answer is A, because essentially, if we went with this one here, right, one parent is 2 small t's, it would mean that they would also be affected, right? But they're not affected. This mom or this dad is not colored in. So it can't be that. The only way to prevent these 2 parents from having the disorder is if they had a capital letter each. Now, what they end up doing is giving their daughter their 2 small t's, and they end up giving her the disorder. And so that's why I would say it's a recessive allele with both parents being heterozygous, because the heterozygous condition here prevents them from having the disorder themselves, right? Now, just remember, everybody, that you can ask questions as I go along, and I will look at the chat periodically. Okay. Oh, what I'm using. Someone's asking me what I'm using. I don't know if you mean right now or in the lessons, but right now I'm just using my good notes, but my lesson videos are made with explain everything. Okay, let's move into the next question. So the table below, let's go over to my highlighter, compares the rate of extinction of mammal species over 2 different time periods. And I think actually this is probably a evolution question. So if you haven't got to evolution, don't panic. I'm just going to do it anyway for anyone who's watching out there and wants to know this. So we've got the rate of extinction of mammal species over 2 different time periods. Okay, so what is the ratio between the rate of extinction from 15 to 1900 compared to 19 to 2000? Okay. So they are trying to get a ratio. Now, I don't know if everybody knows how to do a ratio out there, but it's actually quite simple. And I've heard that they don't teach this in maths, right? Which is quite surprising. They teach it in maths literacy, as far as I know. But basically, you've got 4,5 is to 90. Right? So what you're actually supposed to do is divide it on either side by the same number, and you should get 1 is to, now I don't have a calculator with me, so I haven't planned this out very well to actually figure this out, but I'm pretty sure it is to 2, or it might be more because this is 4,5, so it should be 4,5 into 90 should be to 2, and that's actually just how you work it out. You don't need any kind of fancy maths equipment, like way to work this out, because it's not going to work out that way. And actually, it might even be 1 is to 20 now that I think about it, because 4,5 going into 90 couldn't possibly be into 2. It's got to be into 20. And so it's really simple to work this one out. So please just know that, by the way, you are going to have to do basic maths at some point, right? Next little sub-question. Study the following effects. Okay, so we've got a double helix of DNA will not unwind during DNA replication. Thank you for confirming it is 20, everybody, because I don't have a calculator with me, and I don't think I have my iPad doesn't have a calculator on it. I haven't installed it. Thank you. So the double helix DNA will not unwind during DNA replication. Okay, so we've got will not unwind. I'm just going to highlight that because it seems important. mRNA will not form, DNA replication will not take place, and translation in protein synthesis will not take place. Okay, which of the following combinations of effects will result if the hydrogen bonds in DNA were strong? Now, just remember, in DNA, you've got your nitrogenous base, and then you've got your other nitrogenous base on the other side, right? And then holding them together is a weak hydrogen bond. Now they're saying, what happens if it was strong? So it never broke. So which of these combinations? Okay, so if these bonds over here never broke, what one of the following combinations will result? if they were not. So the double helix will not unwind during DNA replication. Yes, so it's got to have that in it. mRNA will not form. Yes. DNA replication will not take place. Yes. Translation in protein synthesis will not take place. I'm going to say yes to that one too. It is all of them actually, because if you think about it, if the hydrogen bond doesn't break, it won't unwind. If it can't unwind, you can't make mRNA, because remember, mRNA is based off of when the DNA pulls apart, right? And you can make a copy. DNA replication will not take place. Of course it can't, because if you can't pull it off, then yes, it won't be able to be read. And lastly, translation in a protein will not take place. That as well, because if you can't pull the DNA apart and read it, you can't transcribe it, which means you can't translate it. So I'm going to go for the option of D. Okay. And for somebody who just asked this now, yes, this will be available to watch later. Where can we get the cheat sheet notes? Everybody, the cheat sheet notes are available under your membership page. You click on the membership. Right at the top, it says perks. You say see perks. You scroll down to the bottom and you'll see something like a link to a Google Drive. In that Google Drive is the sheet in both English and Afrikaans. And if you don't get access immediately, it's just because I haven't clicked on yes yet. So I do get a little email saying that you want access. It's just that so many come in through a day that I don't get to see them all immediately. But I try and do them within 48 hours of you asking for access. All right, let's move on to another question. And I feel like this is a dreaded question for everybody. There's a quick question here asking you want to join the membership. You can. The easiest way to join my membership is to go on a computer, actually. Sometimes it's difficult to join on a phone, but basically you click the join button on my YouTube home channel. If you want all the access to everything, you're going to choose the Rescue Me option, and there you will get access to the membership videos that are private, these live lessons that will also be made private. What else is there? There's also this cheat sheet, of course. And then members are allowed to ask me questions as much as they like, actually. And you can ask me questions and I can respond to you as quickly as I can, which I don't do for normal members, or normal subscribers, should I say. All right, Sunita, I know you want to see this question. Yes, it is a nightmare. It's difficult, especially if you don't know what to do. So we're going to practice it now, because you might see something like this in the upcoming paper. So it says, contraceptives are used to prevent pregnancy, and some females use pills that contain progesterone. In one packet, there would be 28 pills. 21 contain different concentrations of progesterone according to the day in the cycle, and the 7 remaining contain no progesterone. A female has to take 1 pill a day at the same time, and a sequence is shown. So there is the sequence. The graph below shows the difference in the progesterone levels during a menstrual cycle of a woman taking contraceptive pills and a woman not taking. And that's also important. There are actually 2 women on here. So I'm actually going to highlight them differently so I can see them more clearly. So this is the woman without. And then this is, sorry, that's a woman with. This is the woman without. Okay. And it's showing the difference in the progesterone levels, right? Okay, so already, before we go any further, I hope you can already determine sort of what's going on here. If you take progesterone pills, your progesterone level peaks, remains stable, and then drops off. That is because you want to prevent pregnancy. Remember, you can't be pregnant if you're already pregnant. I mean, so you can't get pregnant, and that's how the contraceptive pill works. Progesterone maintains a pregnancy, right? So if you keep the level high, you fool your body into believing you're pregnant. Therefore, you cannot fall pregnant. Versus this woman over here without the pills, she has a low level of progesterone, which means that she can fall pregnant in this window over here, especially over here, because that's where she ovulates. Then from that point, her progesterone levels go up in case she is fertilized, and therefore the pregnancy can be maintained. But as we can see here, the progesterone levels drop off, which means that she didn't fall pregnant. Otherwise, if she did fall pregnant, this would continue straight, but it doesn't, it goes down. So let's get into the questions. Number 1 says the estrogen levels between day 8 and 22 will remain low in the will remain low in the woman who takes the contraceptive pills. Explain why this is the case. So let's go to day, what does it say? The estrogen levels between day 8, so it's here. Oh, yeah, day 8 and 22, there we go, will remain low in the woman who takes contraceptive pills. Explain why this is the case. Now, remember, we're focusing on estrogen here. We're not talking about progesterone, right? Okay. So, this is a lot to unpack. Estrogen, everybody, is for what? I'm just going to scribble down my answer here. Let's remember that estrogen does what? It replaces the endometrium, right? Because it's fallen away. Now, why would that level be low? Because that's what it's saying. It's saying the estrogen levels between 8 and 22 will remain low in the woman who takes contraceptive pills. Explain why this is the case. Now, it's for 4 marks, so you have to say quite a bit. So the first thing that you need to acknowledge is, number 1, what does estrogen do? Estrogen replaces the endometrium. Tick. However, right? And so that's the point here. This is a however sign. Okay, however, if she's taking progesterone, we don't need estrogen because her body thinks it already has an endometrium layer, right? So now you have to explain that, well, estrogen is secreted to replace the endometrium. However, if there is progesterone being released, the endometrium will be maintained. Okay. And if there are high levels of progesterone, there will be lower levels of estrogen. Why, you say? Because if progesterone is high, then you won't be making another follicle. And if you won't be making another follicle, you won't be making estrogen. Now, if I've lost you, let me just take you a quick step back. Remember that we secrete FSH. FSH goes to the ovaries and we mature a follicle. Here is my follicle. The follicle secretes out estrogen. If progesterone is high, we won't secrete follicle-stimulating hormone. And because we won't secrete follicle-stimulating hormone, we won't make a follicle, which means we won't make estrogen. Are you following with me? Why do you say? Because we won't want to make another follicle if we are already pregnant. Now, remember, we're pretending to be pregnant because, of course, we're preventing pregnancy. So if progesterone is high, okay, there will be no need for estrogen to replace the endometrium because we are inhibiting FSH, and by inhibiting FSH, you are inhibiting the production of a follicle, and if you inhibit the production of a follicle, you inhibit the secretion of estrogen because estrogen comes out of the follicle. And therefore, we have a lower level of estrogen and we maintain our progesterone level. Even though progesterone and estrogen are hormones that sort of like work together with one another, they ultimately do sit on a balance scale. In other words, you can't have too much or too little of a substance. All right, so that's quite a bit to cover in those 4, but you would essentially get 1 for speaking about replacing the endometrium, acknowledging what estrogen does. Then you're going to speak about the fact that if progesterone levels are high, FSH won't be secreted, therefore a follicle won't be secreted, therefore estrogen will be low, will not be secreted. So there's your 4 marks acknowledging those 4 things. Okay, this was a tough one. There's probably some alternatives that you can also speak about here, and I just want to remind you that the memos online for these past papers are not whole, if that makes sense. Sometimes what you don't see online is all the extra bits and pieces that are added, but you never get to see those because those are, let's say, not a secret, but they are kept at the marking centers. So we never get to see the alternate answers that were approved as well. All right, let's move on to our next question. It says ovulation took place on day 14 in the woman not taking the contraceptive pills. Explain the evidence in the graph that supports this conclusion. Now, I want to remind you that this is for 2 marks. It's an explain question, and I say this to my students all the time. If it is an explain question, it must have a statement and a reason, right? So statement, reason. And you could have actually done the same for this one too, except this one would have had 1 statement and 3 reasons, okay? That to get to your 4. So, statement, reason. We know that ovulation took place on day 14, looking here, because look at the spike of her progesterone level. So it says, explain the evidence in the graph that supports this. So, statement, right? The progesterone increases on day 14. That's the spike. We can see it on the graph. That's your statement. Okay. The reasoning is because of why. Progesterone needs to increase in order to maintain and prepare the endometrium for implantation. You could go just a little bit further under the word prepare and say something like to make it more glandular and vascular. So you could say progesterone increases on day 14 as seen in the graph in order to maintain and make the endometrium more glandular and vascular so that implantation can take place. And that's all you're going to say for that to get 2 marks. Okay, next one. Suggest. Now, this is a higher order question, so it actually requires you to think a little bit beyond what's in the textbook sometimes. This is a higher order level question. There aren't a lot of these in exams generally, but it says suggest 1 reason for including pills with no hormones in the contraceptive packet. Now, if you go back up to the top, you will see, yes, there are some pills over there. Now, this is interesting because it says suggest, so you've got to come up with your own answer. And probably the majority of people would write, and it's not wrong to write this, would be something like to allow for menstruation to occur, right? Maybe something like that. In other words, not to allow the woman to like menstruate, to bleed, to have a period. You could say any one of those words would be applicable. And then for her to grow a new lining the next month. In other words, to follow a normal cycle. And that's all you have to say. You don't need to elaborate any more than that. It's just suggesting a reason. Now, the last one, which I'm just going to clear up this area over here so I can work down here because I'm going to have to draw some things, is, describe the development of a zygote until implantation occurs for 4 marks. Now, for a describe question, everybody, it's not just a statement and a reason like explain. What is it? That is where you need to provide things like how it happens, where it happens, why it happens, when, and sometimes what. And the what and the how are sometimes linked to one another as well. So they're sort of interchangeable. Now, this is for 4 marks. So we need to say 4 things. So we need to start at zygote and we need to end at implantation. So that's point A to point B is in our answer, right? So here we go. If we have a zygote is a diploid cell. Now it needs to undergo mitosis, right? In order to form a morula. It undergoes mitosis even further to form a blastocyst, right? So effectively, we go from one cell to a ball of cells that is solid to a hollow ball of cells. And the reason why it becomes hollow over time is because this is going to be the internal cavity of our embryo in the beginning, right? So it's asking you to develop a zygote, which we have, here's the zygote, to implantation. So what are we going to say? The zygote undergoes mitosis to form a morula. The morula continues to undergo mitosis to form a blastocyst. We're not done yet. The blastocyst, and now we're just going to draw a little wall here. This is the endometrium wall, and then this is the smooth muscle underneath it. The blastocyst, there's our blastocyst, grows what we call chorionic villi, which are like these little extensions, like that, into the wall of the uterus, into the endometrium of the uterus, and implantation has occurred. And now we are at implantation. So you're going to get marks for mentioning the structures and the process, mitosis. And that's going to get you 4 out of 4. And that's why we need to know this process very well. I have included this in the cheat sheet as well for everybody to use. Now I'm going to take a quick pause. I'm going to show you the next question we're going to do, which is a meiosis question. So you can just have a look at it while we wait. And I just want to see if there are any questions in the chat that I'm going to open up now. And then I can just have a quick squiz at them. I can do a little Punnett square for the request there about when can we do a Punnett square. We can do one a little bit later. When will I be revising the eye? I will probably be doing some more paper 1 next week. Not today, though. I'm doing a variety of questions today. Let's see what else the live chat says here. Let's see. This video does go onto my YouTube. But this video goes under the members section. Unfortunately, it's not saved anywhere else. If you are obviously a member, then you will see this. And can I repeat the development of a zygote? Sure, I can do that for you. Okay, so a zygote. This is a zygote, which, by the way, I left the part out about how to make a zygote because I assumed everybody maybe knew, but just to remind you, this is my egg cell, which is haploid. This is my sperm cell, which is haploid, right? They come together via fertilization and they make a zygote. The zygote, which is now diploid, undergoes mitosis, so the cells double and triple and quadruple, and they make a morula, or a solid ball of cells. The morula continues to go under mitosis. This forms a blastocyst, which is a hollow ball of cells. This will eventually become the outer layer of the embryo with the inner organs growing inside here. Okay, so this blastocyst moves down into the uterus. Here is my uterus wall, and here is my endometrium, the squiggly line. The blastocyst grow chorionic villi, which are like these long finger structures that attach to the wall of the endometrium, or to the actual endometrium. They grow in, and now implantation has occurred. Okay, that's my little recap for you. Okie dokie. All right, let's move on to this because we've got two other questions I want to do, another one on DNA as well that I want to get to, and translation and transcription. Okie dokie, so this one is about meiosis, and please don't underestimate how difficult meiosis questions can be. I always warn my grade 12s that they can be very difficult, and we don't put a lot of effort into studying meiosis, and then we write our mock exam, or even our final, and we're like, whew, that was much harder than I thought. And they make it difficult because the content is easier, so they have to make a harder question, basically. So it says diagrams 1 and 2 represent gametogenesis in a human male and a human female, not in any particular sequence. Okay, so the diagrams are not drawn to scale. That's fine. And it's basically breaking it down here, what it looks like in these two individuals. Now, before I go any further, I can already see which one is male and which one is female. And I'm hoping that some of you can also see. But if you can't, let me tell you, the key is to look down here. That's the key to telling us whether it's male or female. In male gametogenesis or male meiosis, you end up with 4 completely identical cells. In females, however, you end up with 3 polar bodies and 1 ovum. And so that's how I know diagram 1 is female. And I'm going to write that down because we probably will need it later. Oh, bless me. And diagram 2 is of a male. Okay, right. Now, let's get into our question. Identify the specific type of gametogenesis in diagram 1. And what they mean by that is you can't just write gametogenesis again, right? You have to write what type of gametogenesis specifically. Well, now that we know that this is female and we've identified this as female, and that's basically what they're asking you to do before answering the question, is we now know that this is going to be oogenesis or oogenesis. Doesn't matter how you pronounce it, as long as you spell it correctly. That's all that matters. Number 2, explain your answer to question 2221 by referring to a visible difference between diagram 1 and 2. This is nice and great. So remember, for all explain questions, to get full marks, we want to give a statement and we want to give a reason, right? So explain your answer by referring to a visible difference. So statement. Our statement could be, let's think, our statement could be about the number of gametes, right? So my statement will be something like females produce 1 ovum and 3 polar bodies, right? Males, as my contradictory or my reasoning, males, on the other hand, produce 4 identical cells. And that's basically all you need to say. That's how you're explaining your answer. Your answer is simply that females, I know that this is oogenesis because females produce 1 egg and 3 polar bodies, whereas males produce 4 identical sperm cells. And that's all you need to say. You don't need to go in any more detail than that. All right. Next question. Where in the human body does this type of gametogenesis shown in diagram 2 take place? So they're talking about this diagram up here, which is in males. So hopefully we all know that answer off by heart. It is the testes. Right. Next question. That was a short and easy one, right? Oh. There we go. Next one. Give the chromosome number of cells at 1. So let's have a look here. Cells at 1. So these cells over here, this cell and this cell. What is the chromosome number? Well, they've undergone meiosis, right? Which means that they have meiosis 1, which means that they have already halved their number. And to go back here, it is in humans, which means when we started this process, we were at 46 up here. After meiosis 1, we're already at 23 chromosomes. And remember, after meiosis 2, we will still be at 23 chromosomes. So my answer for this one is going to be 23. If we go to cell 2, cell 2 is also 23 chromosomes. Now, I just want to warn you, and especially if there are some people in the chat that are rewriting their exams, you can have 2 answers that follow on one another like this. In other words, what I mean by that is, don't be afraid that they've asked the same thing twice here, like you've written the same answer twice. That can happen. And unfortunately, what they're trying to do is they're trying to test your confidence. How confident are you in your 2 answers here? Because sometimes you're not confident enough and you go, ooh, is this possible? Would they really ask 2 answers that are the same, one after another? The answer is yes. And they do it all the time. They're checking your confidence and whether you trust your ability to calculate things. So the answer is 23 and 23. All right. 2225. Name 2 processes that take place during meiosis 1 that lead to genetic variation in the 4 cells shown at 3 in diagram 2. So we're just going to go over here. So we're talking about males over here. So what are our 2 processes? Well, my answer for that would be, and it must be during meiosis 1, hey? I would say crossing over, because remember, crossing over can only occur in meiosis 1. Remember, it cannot occur in meiosis 2. And then the second one would be random arrangement. Okay. And that's really it. That's all you need to give. We don't talk about independent assortment right now, and I would suggest not giving that answer. I also wouldn't give mutation either. Okay. Sangeeta wants to ask, ma'am, on explain questions, how do we make the statement and how do we give a reason for a particular statement, and do statements have to match the reasons always? So I'm going to explain that when I'm done with this very last question, and then maybe I can do it while I do the explain bit for you now so you can see it in action. Okay. So this next question is an explain question. And the reason why I put so much emphasis on how to answer explain questions correctly is because often these are the ones you don't get full marks for. And then you wonder why you get like 60% in the exam instead of getting 90 or 80%. And it's probably because you're not correctly answering the explain question. So it says explain the implication for a human. Implication means like outcome or result. For a human population size, if the 3 cells referred to in diagram 1 did not degenerate, but remained as gametes. So if we just go back up here, we look at this diagram over here. What they're saying is, what would happen to the human population if these 3 cells were all eggs as well? So we had 4 eggs instead of just 1. Basically, what they're saying is, if women were making 4 eggs a month versus 1 egg a month, what are the implications on the human population? And hopefully you can all guess. Basically, it would quadruple our population. We would go from having 1 baby per woman to potentially 4 babies every time from every woman. That's basically what they want to get at, right? So if we just get rid of this, so I can write my answer down here nicely. So let's talk about, because I'm going to answer Sangeeta's question about explaining, right? So explain for 2 marks, okay? Statement, reason. To make it nice and easy, I want you to think of the statement being a sentence that you're going to show the marker you understand the process or the fact or what's being asked, and then you're giving a reason to how you know it is true or it's a fact. And so that's why these two must match one another, right? The statement is often a statement of a fact, and then you are supporting it with a reason as to why that fact, that statement is true or correct. So what is the implication if 3 cells did not degenerate? So statement. Females would have 4 egg cells available for fertilization. So that's a statement. She would. That's a fact. If we go back to the picture, fact. If all of these grew to full size, we would have 4 eggs instead of 1. So that's my statement, my fact. Now, the reasoning behind this is to support what is going to happen. It's going to give us the second part because it says, what is the implication of this for the human population cells? Well, if females had 4 eggs for fertilization, then, and that's what you can sort of think in your mind, the reason is the then. It's almost like this is the if statement, and then this is the then reason. So if females had 4 eggs available for fertilization, then 4 babies would, or maybe we can even say could, be born each time, increasing the population by 4 times. Okay, I hope that helps you break down how to give statement and reasons for explaining questions. And to take it just one step further, if this question was out of 3, then you must give a second reason, and then that's what would give you your 3 marks. This one doesn't have a third reason, so we're not, or second reason, so we're not going to add that in. I hope that answers your question. All right, now we're going to go into a DNA question. I'm just going to leave this up for you to have a look at. And I'm going to have a look at the chat now for everybody so we can see if there are any questions. So Sangeeta, I hope that has answered your question. Pravina, meiosis is most definitely a nightmare. I think it is a nightmare for everybody. And if you've never written an exam with meiosis in it yet, you don't know what's waiting for you on the other side, but sometimes it's very challenging. All right, let's have a look at some of the questions in the chat. Nicole is asking, how do twins and triplets occur if there's only one egg cell? I can just show you, let's see, I don't want to draw it on here, but it's very simple. I'm just going to zoom in super close so I can draw it for you. But basically, twins just occur because if you have one egg and you have one sperm cell, what happens is after fertilization during mitosis, the egg cell splits into 2 by accident. And then you end up with one twin and the other half of the twin. The other thing is you could get 2 egg cells and 2 sperm cells, and then you can have non-identical twins. And the same thing happens with triplets. You get non-identical triplets and you get identical triplets. If they're identical, they've shared an egg. If they're non-identical, they haven't shared an egg. Okay. Right, I'm just going to leave the DNA question. This is DNA and like translation and transcription and mutations all at the same time. So I'm pretty sure you want to see me do this one. I'm quickly going to look at the chat. This lesson is being recorded, everybody. I actually have done a reproduction question already for someone who's just asked that now in the chat. It's earlier on in this video, so the video is going to be on my YouTube after this. It's all recorded and it uploads the moment the lesson is done. It takes a little while because it is a live video stream, so it's a big file, but it does appear on our community page as well. All right, so let's get into this DNA question. All right, so it says the diagram below show or represents nitrogenous-based sequence of a part of a nucleic acid. They don't tell us which nucleic acid. That codes for making insulin. Insulin is necessary for the uptake of sugar in the blood, blah, blah, blah. Okay, the sequence below are 2 individuals and are read from left to right. So we always read from left to right. Sequence 1 is our normal person, and sequence 2 is from a person who shows a mutation and cannot produce insulin, which probably means they're a diabetic, because as we know, people who don't make diabetic. Oh, you want me to zoom in? I can zoom in. Sorry, is that better, everybody? I was just looking to see the comments. There we go. Okay, so now we've got our sequence of DNA, and we now need to see where the mutation has. Now, I don't read the questions yet, and I never do, and I tell my matrics not to do the questions first. In other words, just really digest what's happening here and find where the code has changed. So I'm going to compare the codes, reading left to right, and already the first sequence is problematic, okay? Look here, it's already changed. This is different to sequence 1. Same, 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 same. So it's this one over here that's changed. And I've highlighted it because it's obviously important. At some point, they're going to ask me that, right? So the first question says, which nucleic acid is represented in both sequences? Which? Now, you've only got 2 options of answers here, okay? Your answers are either going to be DNA or RNA. Those are the only 2 kinds of nucleic acids that you can get. Obviously, there are versions of RNA, like mRNA and tRNA, but these are the 2 main kinds. So which nucleic acid is represented here? I feel like you should know by seeing the fact that there is the letter T present here, right? And the letter T can only be found in DNA. So the answer is, what is the nucleic acid? It is DNA, so it is definitely not RNA. T is for thymine, which is only in DNA. All right, let's move on to our next question. Where did the mutation in the second sequence occur? So if we have a look up here, where did it occur? Now, you've got to think, where did it occur? What are they talking about? Like, where is the DNA? Okay, where did the mutation occur? What you can acknowledge here is two things. You can say that the mutation occurred in the nucleus, and now you must acknowledge the mutation where TAG became TAT in the first DNA triplet. First DNA triplet. So that would be my full sentence. I would say this. I would say the mutation occurred in the nucleus where the base triplet was TAG and is now TAT, changing the first DNA triplet. I know it's quite a long sentence for 1 mark, but what you're doing is you're making sure that you've covered all your bases when they say where. Because remember, you don't have the memo in front of you. You don't know if they're trying to focus on the location within the cell, the location within the DNA. You're not sure. And we don't have the memo in front of us, so we can't compare all the time because obviously you won't in an exam. So cover your bases and tell me where it's happening in the nucleus, where it's happening in terms of the letter change in the sequence, and where it is physically in this table. It is the first DNA triplet. So you've covered all your bases by putting this in all one sentence. Now it goes on to say, it's a follow-on question. The table below shows the amino acids coded for by the different DNA base triplets. So I just want to acknowledge here that this is DNA triplets. They're not mRNAs like we're used to seeing. Okay, that's important. Normally, this is an mRNA table. This is now a DNA base triplet. It says, for our follow-on question, give the anticodon of the fourth base triplet on sequence 2. So let's go up to sequence 2, to the fourth base triplet. We'll highlight it in a different color. Sequence 2, number 4. 1, 2, 3, 4. Yeah. Right? So the DNA sequence is GTT. Let's just scribble this down here so we can do the calculations. So it's GTT. That is the DNA base triplet. But they're looking for the anticodon. So let's do the working out. Let's not skip any steps. And by the way, any of you who want to skip straight to the anticodon without doing the working out, I don't suggest that, even if you are the smartest student, because there's always room for error when you are writing exams and you are nervous. So rather take it slow, do the working out, and now you will get 2 out of 2 and you won't sacrifice losing silly marks. This is our DNA sequence. Let's make the mRNA sequence. Right, remember there must be complementary codons, complementary nitrogenous bases, keeping in mind that there is no T's in mRNAs, right? So the complementary code should be CAA. Now, if we're going to take it to the tRNA, we are going to then have to make this complementary, which is G, yes, U. Okay, that's going to get you 2 marks. And yes, it's taking me a little bit longer because I'm saying it out loud to you, but look how quickly you can quickly scribble this down in pencil on the exam paper and get the right answer and not sacrifice easy marks. Next question says, give the amino acid coded for by the first base triplet in sequence 2. So now we're going to go back up and it says the first base triplet, which is this one. So that's TAT, right? So we'll go back down here and it says amino acid for TAT. So we've got the first base triplet is TAT, and now we want the amino acid. So we just go back up to our list here and we look for TAT, which is over here. There it is. So our amino acid is isoleucine. And that's what we write there for 2 marks. And what I would do for that is I would make sure that you write out TAT and then you write isoleucine to get 2 out of 2. I would write it like that. And again, that secures your marks. And so you don't have to, panic that you're going to miss out on anything. Now, this is a nice, simple DNA question, so it's not too challenging. The one thing I want to say to everyone before I go on to our last little section for the day, I think there is one more question after this, although I could be wrong. But please always make sure that if the base changes, please actually make sure it makes a different amino acid. Like, please confirm that, okay? So what I mean by that is there isn't a TAG on this list. If you compare it to the original sequence, there is no TAG here. But I need you to make sure that if they ask you any question in the exam, please make sure that it actually changes it. So what I'm getting at is, do you see that there are like 2 codes for each amino acid here, but their lettering is different? If they say in the exam, let's say, zoom in here, let's say the code was GTA, but now it has changed, it's mutated to GTG. What is the effect? Please make sure that you check what is the actual effect, because as you can see here, the effect is nothing. They both make histidine. It doesn't matter if the letter is mutated and changes from A to G. It's still going to make histidine. So please, please make sure that you double check that. Okay. I think that's my last question. So what I'm going to do is I'm going to just open the floor for requests. I know somebody wanted to see a Punnett square, so I'm just going to open up my little chat. Sangita, you want me to do 2, 3, 3 again? I can. 2, 3, 3. The whole thing. I'll do a quick sticks. So give the anticodon of the 4th base triplet on sequence 2. So we went up here. Here is my 4th base triplet. So we highlighted. It's GTT. So I wrote down GTT. That's my DNA base triplet. I now wrote down the mRNA, which is complementary to this. So guanine is complementary to cytosine, and thymine is to adenine. So it's AA. I then need to make the tRNA, and tRNA is complementary to mRNA. So G joins with C, so I write G. A joins with U. Now, why am I using U? Because remember, in RNA, there is no T. So just to remind you, in DNA, T joins with A and G joins with C. But in RNA, U joins with A and G joins with C still. Okay, and that's how I got that answer. And then for the second one, give the amino acid coded for by the 1st base triplet. Well, the 1st base triplet was TAT. I just simply go up to my table. I find TAT, which is here, and that gives me isoleucine. Okay, right. Let's have a quick look at the chat. I know there was a genetics question. I see you. Don't worry. Oh, you want me to, the making sure thing, not to corso. I see it. Oh, the making sure thing. I'll say it again. If in an exam, they say, this was the original code. Oh, whoops. They say the original code is GTA. They then say a mutation occurred. The code is now GTG. That's the new code. This is the question, what is the effect on protein synthesis? Well, the effect is, yes, the baselet triplet has changed, which means the mRNA will be different, the tRNA will be different. However, the amino acid will still be the same, because even though the base triplets are different and therefore their mRNAs will be different and their anticodons will be different, they will still bring the same amino acid. Therefore, there will be no change in the protein that you make at the end. The process was different, but the result is the same. And they like to catch you out on that, because if you don't read your table well enough and you don't notice that these two actually code for the same amino acid, even though they have different letters, you are going to get caught out in the exam. So double check, please, that if they ever ask you a mutation question about changing any of these letters, or changing the mRNA, or changing the tRNA, whatever the lettering that's changed, make sure, does it actually change what I'm making? Or does it change the letter, but not actually the amino acid I'm making? Because many base triplets, many codons code for the same amino acid. We can see that here for glycine. There are 3 base triplets that all code for glycine. So even though these have changed, and maybe that's what they're trying to say, they're trying to say, well, if there's a mutation, does it matter? Well, it matters in the sense that, yes, it's a different mRNA that will come. But at the end of the day, you'll still bring the same amino acid, which means you'll still make the same protein. So you've changed the mRNA, but you haven't changed the protein or the amino acid, the product at the end. That's what I'm saying. So the letters sometimes might be different, but the outcome is exactly the same. Okay. Right. Now, I know somebody asked for a Punnett square, so I'm just going to ask what kind of Punnett square, because there's more than one kind I can do. And I just want to clarify that I'm probably going to then have to do a monohybrid, and maybe if I can do a dihybrid too, I can kill 2 birds with 1 stone in the same question. But let's just quickly do a dihybrid, because it's the same as a monohybrid in the layout. It just takes longer. And by the way, the good news is, everybody, is that you'll never have to do a full dihybrid in an exam, just so that you know. You'll never have to fill out a full 16 squares. So let's just do a dihybrid, right? Quick. Yeah. So let's say we have, let's do something. Let's do a classic. So we've got tall, and we've got short. And then we've got, let's think. Let's do white. Oh, why am I writing that out? No, W is not a nice letter. Let's do black as a better letter. So B for black, and then little b for white. So hopefully we already know capital letters mean that's the dominant allele, and smaller letters mean that's the recessive allele. So let's say the question says like it's 2 heterozygous parents. So the parents are this, basically. That's what a heterozygous parent looks like. Okay. Oh, why is it jumping between the 2 pages? There we go. Okay. So in the margin here, I go P1, and those are my parents. And then what we now need to do is we need to make some Punnett squares, and we need to finish this out properly. Please remember that the format of this is really important. So we've got P1, those are our parents. And now what we need to do is we need to go gametes in the margin over here. And we need to go meiosis. Okay. Now what I tell my matrics, so that they never get this wrong, is I like to draw circles to represent the cells you're making. So for each of these people, there must be 4 cells. In a monohybrid, there would just be 2 each. This is my genotype, everybody. Can I just take a quick second here to say something which is bothering me quite a bit? I've seen a lot of stuff on social media and on TikTok too, other biology teachers teaching this section. And they write a lot of stuff that doesn't matter, or quite frankly is wrong. And it's very frustrating, because I can see a lot of people enjoy watching these other biology teachers, and I'm not saying that they are wrong. I'm just saying they write a lot of stuff that's not important or that's not going to get you any marks. And people are like, well, ma'am, you haven't written the genotype here. I have written the genotype. It's up here. If you're asking me, why didn't I write the word genotype over here? It's because you don't have to. It's not going to get you any marks. It's not important. And if you ask me, okay, ma'am, but how do you know that? Like, I'm scared. I'm not going to get marks. Everybody, I mark final exams for paper 2. This is the kind of question I mark. And I'm telling you, it doesn't have to be there. It is not on the memo. It is not awarded any marks. So I'm saying all you have to do is write the genotype of the parents. I'm actually going to write the phenotype as well because I forgot to put that at the top here. So this is a tall and black parent, and this is also a tall black parent. But you don't have to write phenotype, tall and black. Genotype, you don't have to do that. You just have to write it, just write it here. You don't need any descriptors along the side here. The only descriptors you need are this P1, gametes, and when we get to F1. Okay, I want to move on from saying that now and go into making these gametes, because this is where everyone makes mistakes. This is what I teach my grade 12s. You can only use each of these letters twice. So what I teach them to do is underline them to show that they've used them once. And also remember the law of independent assortment and the law of segregation is really important here now. So the law of segregation says that you can only inherit an allele twice from each parent, no more. So what that means is, for my first one, I'm going to use this letter T and this capital B, so I put it in there. I can use this capital T one more time, so I underline it to keep a track of it, but now I use this B, little b. I am also technically, if you do maths, I'm foiling it out, which means I'm going this T times this B, this T times that B. I'm using the idea of foil. Now we go to the next one. I'm going to use my other T, and I can use the capital B. Now I've used that B twice, so I can't do it again. And then I'm going to use T and B. And now I've got my gametes. Now, I'm not going to waste time doing these because do you see that this genotype is the same as this genotype, which means their gametes will be exactly the same. So I'm just going to copy them over from this side, like so. Right, now we scroll up. We go fertilization. That's important. That must be there. We now go in the margin here, F1. And now we draw our enormous Punnett square. So I'm going to just zoom out a bit. One, two, three, four. Here's the fourth one. I think that should be enough. There we go. Okay. So now we're going to copy in my gametes. So we've got there, 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 and there. And then it's exactly the same going down the side because the parents are the same. Okay. Now, somebody asked in the comments, I think it was Sunita, can you do like foiling? Yes, so foiling basically means this block times this block times this block. So we're doing this kind of thing. That's foiling, okay? And that's basically what I'm doing. So this block times this block means I've got 2 capital T's, 2 big B's. Then I go over to the next one, 2 capital T's, a big B, and a small b. And then the next one, capital T, lowercase t, 2 big B's. Again. Right, move one down. T times T, 2 big T's. Big B, small b. And yes, you should always write the big letter first, everybody. 2 big T's, 2 small b's. Big T, small b. Big B, small b. Big T, little t. 2 small b's. Question just asked, can you write it like this? I didn't see the person's name. They can write like this. You can. It's not wrong, but I wouldn't. It's a very bad idea because how are you supposed to read this? And what I mean by that is you're supposed to put the letters that are alike with each other. It's supposed to be the two T's together with the two B's together. I wouldn't do this, no. You will run the risk of getting your whole Punnett square wrong if you make a small mistake like that. So I would not do that. No, that's not my suggestion. It's a strong suggestion not to do that. Right, let's keep multiplying it into the Punnett square. So we've got capital T, little t, big B, capital T, little t, big B, little b, small t, big B, small t, big B, little b. Last row, big T, B, big T, little t, B. Two little t's, big B, small b. Two little t's and two small b's. Whew, it's a workout to do this whole thing. Okay, so there it is. Now, this is why you should really have some highlighters with you in the exam. I'm going to do the phenotype. In other words, I'm going to go around here and put everybody who is the same color and size together, starting off with the fact that we've got tall and black. So I'm looking for all individuals that are tall and black, which means they need at least one capital T and one capital B. So tall and black, yes. 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 Tall and black, no. Just tall, that is white. Tall and black, yes. Tall and black, no. Tall and black, yes. Tall and black, yes. Tall and black, no. Tall and black, no. Oops. Last one. Tall and black, yes. Tall, yes, but not black. Tall and black, no. No. So I should have 1, 2, 3, 4, 5, 6, 7, 8, 9. And I know that I'm correct because this is a part of our ratio that we must know off by heart. So these are all tall and black. All right, let's do the next one. Let's go tall and white. So tall and white, yes. Tall, white. Tall and white, yes. Tall and white, no. Short and white. Tall and white, no. Tall and white, yes. Tall with white. Tall and white, no. Tall and white, no. So there's the next part of our ratio. It's 3 out of 16 are tall and white. Okay, next one. Let's do a different color. Let's do now, because we've done the talls and the colors, now let's do the shorts and the colors. So let's do short and black. So short, yes. Black, yes. Short and black, yes. Short and black, short, yes. Short and black, no. So again, we have 3 out of 16 is short and black, which leaves us with our last little person over here, which is 1 out of 16, which is short and white. And that gives us that golden ratio of 9 is to 3 is to 3 is to 1. And this ratio is golden because if they ever ask you, what were the parents, without showing you the parents, you can only get this ratio, grade 12s and my rewriters, you can only get that ratio if the parents are heterozygous. Both of them need to be heterozygous. That's the only way we get this golden ratio. Now, the good news is, right, what's the good news? You'll never have to do a whole Punnett square like this. They often just have like one of these missing, and you've got to fill in the missing one. The other questions they ask is, they have a whole Punnett square, but like one of the gametes is missing. And so they ask you, what is the gamete that should be here? And you can work it out just by looking at the answers in the row. You can figure it out. The other question is sometimes like, these are the gametes, right? What are the parents? And so you've got to work backwards by taking these gametes and putting them back together to their parent genotypes. And so the good news is you don't need to ever do a full one of these. Your teacher might make you do a full one in class, and I make my own students do a full one in class to practice. But the good news is you don't ever have to do that in the final exam. Right, I'm going to look through the questions here. Somebody asked what was the percentage. You don't have to do percentages, by the way, unless it asks for it. You can do fractions. 9 out of 16 is okay, but 3 out of 16 as a percentage will give you a weird percentage. So you don't have to give percentages, everybody. You can give fractions. But if they want a percentage, you can then give it to them. But that's why you'll need a calculator. Okay, to answer that last question, yes, this whole video is going to be available up on the membership video playlist. So go and have a look there. And that's it for today's live lesson. We did a lot. We've been busy for over an hour now. For those of you who are looking for some more live lessons, please keep an eye. I'm going to do another one next week. Then I'm going to do some other kinds of questions, specifically for people who are rewriting. So I'm going to do some eye, some ear. I'm going to do some endocrine system ones. Keep an eye out for that on your community page, because that's where I put the link. Sangita asks, does the circling for the gamete only apply for monohybrids? No, you can do the circling too. You can do it in any of the mono or dihybrid. I just put the circles so I keep them separate. But if this was a monohybrid, it would just look like that. I would have 4 circles instead of 8. Okay, thank you everybody, and please go and join the membership page if you want to. Don't forget you can get my study guide, which has all of this in there, and also there's a whole bunch of extra videos that you get access to as a member too. And I will see you all again soon, everybody. Bye.